Welcome back. Last time we talked about the events of mitosis and cytokinesis, and what we're going to move on to now is to look at meiosis. So there are some significant similarities between the events of meiotic cell division and mitotic cell division, but there are also some very important differences. So I'd encourage you to think about the similarities, but also to notice the differences when they come up, and I'll try and point those out as we're going along. So let's just start off by taking a look at the functions of meiosis. Meiosis basically has one function, and that is to produce sex cells. So those are the gametes, and those are either sperm or egg. So that is the only function that we need to worry about. So before we get going, taking a look at meiosis, let's do some questions just to kind of build a little bit of a framework around which we can hang our new knowledge of meiosis. So I'm going to read through these questions and talk about them a little bit. Half the genes in your somatic cells come from your mother, half from your mother, half from your father. So that is true. You get half of the genes from your mom and half the genes from your dad. So your relatedness to both your parents is 50% or 0.5. And that's because of what's going to happen in meiosis. And so we'll be able to explain that by the time we're finished with this discussion of meiosis. Second one, half the chromosomes in your somatic cells come from your mother, half from your father. Also true. And so we will look at the behavior of the chromosomes and see how they basically end up half coming from mom and half coming from dad. And that means at the end we'll have to take a little bit of a kind of sidestep and look a little bit at sexual reproduction, which we can do once we finish our discussion of meiosis. Okay, how many chromosomes are there in your somatic cells? This question's a little bit difficult. Let's just talk about somatic cells really quickly in case you're distracted by that word. Somatic cells are any cells which are not your sex cells. So anything which is not going to give rise to your sex cells, the germ cells, or the sex cells themselves. So, you know, the skin cells, heart cells, muscle cells, lung cells, all those things are somatic cells. So that makes up the bulk of your body. And so we got 23, 46, and 92. So a lot of you all know that the diploid number is 46. Now, diploid means that there are two copies of each chromosome. So the diploid number is 46. So 46 is a good answer, but so is 92. And the reason that 92 is a good answer is that if you think about our discussion of mitosis, before you can take one cell and divide it into two, you've got to take each pair of chromosomes and replicate them. Because as you're diploid, you've got two copies of each chromosome. So when you replicate those 46, you get 92. So a cell in G1 would absolutely have 46 chromosomes. A cell in S phase, as those chromosomes are being replicated, somewhere between 46 and 92. But if you're in G2, just about going to mitosis, for example, then you're going to have 92 chromosomes. So that's a little bit of a fuzzy question, but a good one to talk about nonetheless. So let's think about your sex cells, sperm and eggs. How many chromosomes are there in your sperm or egg? And this starts to get us to the crux of what meiosis does. Let's think about mitosis for a moment, mitotic cell division. That takes one diploid cell and it divides that cell into two new cells, each of which are both diploid. Is what meiosis is going to do is it's going to take one cell, divide it, and then take each of those two cells and divide them again. So we're going to get 4 cells from this, and the point of meiosis is to make haploid cells. Haploid meaning you only have one copy of each chromosome. So that means if you've got chromosome 1 from mom and chromosome 1 from dad, when you make a sperm or an egg, that sperm or egg can either have the chromosome from mom or the chromosome from dad. So you're always going to have 50% of one and 50% of the other, because half the cells are going to have, or all of the cells are going to have some chromosomes from mom and chromosomes from dad. So in your sperm or egg, we're going to go from the diploid number, which is 46, to half of that, the haploid number, which is 23. Here comes Eddie bringing us a toy. Thank you. We don't need that, but we appreciate the effort anyway. Okay, so the haploid number is 23. Stay off the keyboard, buddy. Okay, so last question here. Let's think about how we go back to being diploid. When the sperm meets the egg at fertilization, so sperm, egg, and the sperm is bringing its 23, and the egg has got its 23, when we reunite those and we make what's called a zygote, which is a fertilized egg, which then undergoes a lot of mitotic cell division and differentiation to give you adult forms. You're bringing 23 in the sperm and 23 in the egg, and so that takes you back to the diploid number of 46. And so that organism then grows, becomes an adult, and in its germ cells, or its reproductive organs, we then do meiosis to make the sex cells, the sperm or the eggs, and we go back from 46 down to 23. And then you go through the whole thing. You bring sperm and egg back together, 23, 23, and you're back to 46. So hopefully that starts off, gives us a foundation to think about the actual process of meiosis. So let's take a look at the process as a whole, and then what we'll do is we'll look at the two parts. So the nice thing about meiosis is that you can break it into two discrete parts. Here is the process as a whole. Let me grab a pen very quickly. So here is the process as a whole. So what we're going to do in meiosis is we're going to have two sets of events. We're going to do what's called meiosis 1, I'll talk about that in a moment, and then we'll do meiosis 2. And so we're going to look at the similarities and differences as we go here, and as we look at the mechanism of what's going on here. So the key to meiotic cell division is that we're going to take a cell in G1. So here's a cell in G1. It's going to go through G1, it's going to go to S phase, and so the chromosomes duplicate, so we're in 92, and then we're going to take that cell and we're going to divide it once, and then we're going to divide those once again. And so we're going to do two divisions, but between the divisions there's going to be no DNA replication. So we're going to go 46 in G1, S phase is going to give us 92 in G2, then we're going to do a division which is basically going to give us 46, and then we're going to do another division which is going to give us 23. And so that means we're going to get 4 cells, each with 23 chromosomes. So we're going to split this into meiosis 1 and meiosis 2. So let's take a look at this diagram. At the top here is the cell we're going to start with. So it's the cell in interphase. Here it says G1. And so let's just look at these two chromosomes here. So this is a homologous pair of chromosomes. So let's talk about homologous chromosomes once more, because this is absolutely critical. So a homologous pair of chromosomes is when you have chromosome 1 from mom and chromosome 1 from dad, and they match up. They've got the same genes. There are variations in the gene sequences, and so those are called alleles. We'll talk about those in genetics more. And so when we have chromosome 1 or chromosome 5 from dad and chromosome 5 from mum, when they come together, we call that a homologous pair of chromosomes. So we're looking at one pair of chromosomes. So that makes this a diploid cell, because there's a copy from mom and a copy from dad. That's what diploid means. It means two copies, one from mom and one from dad. That is absolutely critical to understand meiosis. Okay, so what we're going to then do is we're going to go into meiosis 1. So the chromosomes condense down, you form the spindle like we saw in mitotic cell division. And so what we're going to end up with is 2 sister chromatids for each of the chromosomes. So those are the sister chromatids for chromosome 1 from mom, and then the blue ones are the 2 sister chromatids from dad. So we start in G1 at the top there, okay? Individual chromosomes, 1 from mom and 1 from dad. Then we replicate that piece of DNA, and now we've got a pair of sister chromatids of that chromosome from mom, and we take dad's chromosome 1 and we replicate it, and now we've got chromosome 1 and a pair of sister chromatids. So you can have a chromosome which is just one piece of DNA, or you can replicate it and make sister chromatids like this, and that's still called a chromosome, but now it's got 2 sister chromatids. So we've gone into meiosis 1, and we've got a pair of homologous chromosomes, and each chromosome has 2 sister chromatids. Okay, the first thing we're going to do in meiosis 1 is we're going to basically separate the 2 homologs. So what we're going to do here is we're going to divide this cell, and one of the cells is going to get the chromosome 1 from mom, there it is down there, and the other cell is going to get the chromosome 1 from dad, there it is down there. So when those chromosomes line up, and then we divide that cell, one cell is going to get the chromosome 1 from mom, and one will get it from dad. Now you've got to bear in mind, we're showing this with one pair of chromosomes. This is happening with 23 pairs of chromosomes in one of your cells that will give rise to the sperm or eggs. So that's the first thing we do. That's meiosis 1. In meiosis 2, is what we're going to do is we're going to take the 2 sister chromatids in each of those chromosomes and pull those apart. So meiosis 2 separates sisters, meiosis 1 separates the homologs. So that takes one from mum and one from dad and separates them. That's meiosis 1. Then in meiosis 2, we take the sisters and we separate those. And so that's the important difference between meiosis 1 and meiosis 2, along with some other things as well. So, and then at the bottom here, you can see we've got 4 cells, and each one is going to have a chromosome, and it's either got a chromosome from mum or a chromosome from dad, like these ones on the right. Okay, so that's like the very high level overview of what happens in meiosis. Let's look at the mechanisms in a little bit more detail. So here is our cell on the left, and we're just about to go into meiosis 1. We're just going to do meiosis 1 now, and then in our next video we'll do meiosis 2. So we're going to start off in prophase. The stages are the same as you go through meiotic division as they are when you do mitotic and when you do mitosis, I guess. So you're going to have prophase, prometaphase, metaphase, anaphase, telophase, cell division, cytokinesis, and then you're going to go through the whole thing again in meiosis 2, and we'll look at that in a moment. Okay, so there are some important additional things that we need now to add when we look at this in a little bit more detail. So, what's going to happen is we are going to look at 3 chromosomes here. So there are 3 sets of chromosomes here. So 3 homologous pairs. There is one homologous pair. Here is another homologous pair here. And then here is the third. So there's kind of like a small chromosome, a middle-sized chromosome, and a big chromosome. So there are the 3 homologous pairs. So we're going to say that's chromosome 1 from mom and 1 from dad, 2 from mom, 2 from dad, 3 from mom and 3 from dad. They make up the homologous pairs. So during meiosis 1, during prophase, the homologs come together. So the homologs get matched up. And then is what happens is you get an exchange of pieces of DNA between the 2 homologs. So a piece of DNA from mom's chromosome gets switched with a piece from dad's chromosome. That's called crossing over or recombination. And so that starts to mix up pieces of DNA. And this is one of the other points of meiosis, is it creates genetic variation by taking pieces of mom's chromosomes and pieces of dad's chromosomes. So although we say you got half of mom's chromosomes, you could have got a piece of dad's chromosome on mom's chromosome. So yeah, you get half of mom's chromosomes, but you don't really, because they get kind of shuffled up a little bit. So the structures where the homologs come together are called bivalents, and then you get the exchange of pieces of DNA through a process called recombination. There are going to be videos in Canvas for this, and it's important that you watch those videos because, again, this is a dynamic process. The words and the pictures alone don't really do this justice. So we've done the homologous recombination, or the crossing over. And then we go into prometaphase 1, and in prometaphase 1 we've got the spindle again, so you can see the spindle with the microtubules here, the same as what's going on in mitosis. And the spindle microtubules are grabbing hold of the homologs, and so we're trying to grab hold of the homologs. So all of the microtubules from this centrosome we're going to grab hold of a particular kinetochore, and all the microtubules from that centrosome are going to grab hold of the other kinetochore in the homologous pair. Then there's a lot of that kind of dancing backwards and forwards and pushing the chromosomes, and they all line up in the metaphase plate. Okay, now this is critically important. We need to look at where you get the variation in meiosis 1. The point of meiosis is to give you these haploid cells, but those haploid cells need to be genetically variable. The whole point of sexual reproduction, of which meiosis is a part, is to produce genetic variety. So look at yourself, look at your parents, look at your siblings. You're similar, but you're different, and this is because partly of the variability that arises in meiosis. So far, we've looked at one place you get variation. So that first place you get variation is the crossing over. We take a piece of dad's DNA and stick it on mum's chromosome, take the piece from mum's and stick it back on dad's. That starts to mix up the variations of the genes. It's like taking 2 decks of cards. You take 2 decks of cards and you shuffle them into one another and then you split them. You can still have the same complement of cards, but you've got some variation in the decks. So metaphase 1 is what's going to happen is all the homologs now line up. And so you can see that in this case, we've got chromosome 1 from mom on the left. So here's chromosome 1 from mom on the left at the top here. Chromosome 1 from dad on the right. 2 from dad is on the left. 2 from mom is on the right. And 3 from dad is on the left. And 3 from mom is on the right. Red is mom, blue is dad. I know. It's stereotypical, but that's what the book uses, so that's what we're going to talk about. So you got to think about, is this the only way that this could be arranged? And the answer is no, because what we've got here is mom on this side and dad on this side. And so that's one way you could arrange it, or you could do it the other way around. You could do them like this. And then the second chromosome could be like this and this, or they could be like this. And so you've got all these different combinations. Each pair of homologs can be either arranged mom on one side, dad on the other, or the other way around. So that when we go into prophase 1 and we pull the homologous chromosomes apart, one of the homologs is going to go to one end of the cell, so this one is heading down to this end of the cell, and this one from dad is heading to this end of the cell. And so this gives us just merely one way of moving the chromosomes. We could have aligned these in different sequence, in different arrangements, and we get different gametes produced. And so this is the second place that we get genetic variability. And so that way that the chromosomes line up is called independent assortment. And it's going to come up when we talk about Mendel's genetics in a week or so. So the fact that you can line up the chromosomes like this, mom on the dad, mom on the left, dad on the right, that's my left and my right, or you can do it the other way around. That happens completely independently of any other event. So what chromosome 1 does, it has no impact on chromosome 2, and chromosome 2 has no impact on chromosome 1, or chromosome 5, or 21, or 17. And so all of these lining up events are completely independent. And so you think about how much variation you have when you can line up 23 pairs of chromosomes, either like this or like this. Now you add in the fact we've done some gene swapping, some allele swapping when we do homologous recombination. You already start to see a huge amount of genetic variation, and I'll come to the last place we get variation right at the end. So that's 2 places so far. So then we do basically telophase. We form our cleavage furrow, and we make that ring of actin and myosin, which then pinches the cell in 2. The nuclear envelope reforms. But let's take a look at these chromosomes that we've got in this nucleus that's starting to reform. So this cell on the left, I'm going to call that cell A. Cell A is going to be different from cell B. So cell A has got chromosome 1 from dad, 2 from dad, and 3 from mum. You can see bits of blue DNA here and bits of red DNA that have been swapped over during recombination. And this cell over on the right has got 1 from mum, 2 from mum, and 3 from dad. So they are different already, not to even mention the homologous recombination that came up just now. So that's meiosis 1. Let me ask you a quick question. After meiosis 1, are the cells haploid? So is that statement, after meiosis 1, cells are haploid, is that true or false? Okay, that's actually true. A lot of people want to answer that as being false, because when you look at meiosis 1 and the end of meiosis 1, meiosis 1 ends down there, so all of this is meiosis 1. So when you look at this, you're like, well, no, look, there are 2 chromosomes in here, and there are 2 chromosomes in here. And so you're tempted to say they're diploid, that at the end of meiosis 1, you've got diploid cells. Here's the crux of the matter. If you're diploid, you've got 2 chromosomes, one's from mom and one's from dad. That's what makes you diploid. So these cells here, they actually only have 1 chromosome to start with. It's just formed of 2 sister chromatids. And in both the cases here, those sister chromatids, and this will always be true, those sister chromatids are from the same parent, because what we did is we took a chromosome and we replicated it. And so you still only have 1 chromosome, it's just been duplicated. And so you don't have the situation where you have 2 chromosomes. When I say 2 chromosomes, I mean you've got 2 independent chromosomes. Is what you have is a pair of sister chromatids. To be diploid, you have 2 sets of each chromosome, one from mom and one from dad. So the answer to that is true. Those cells are haploid because they've only got 1 chromosome and it's from mom or dad in this particular case. So that wraps us up with our kind of brief overview of meiosis and with a detailed discussion of meiosis 1. Next time, we'll take a look at meiosis 2.